Welcome to our journey through the history of the tobacco mosaic virus, the first virus ever discovered. Our story takes us back to a time when the microscopic world was still shrouded in mystery, long before we understood the true nature of these infectious agents. This groundbreaking discovery revolutionized our understanding of microbiology and virology, paving the way for modern medicine and shaping our knowledge of the world around us. For centuries, farmers had observed a peculiar ailment afflicting their tobacco crops. A strange mosaic-like mottling would appear on the leaves, stunting the plant's growth and diminishing their precious yield. This enigmatic disease, known as tobacco mosaic disease, would become the focal point of a scientific mystery that would unravel the secrets of a hidden world. Join us now as we delve into the annals of history, tracing the footsteps of pioneering scientists who dared to explore the unknown and, in doing so, unveiled the existence of viruses, entities far smaller and more intricate than anything they could have ever imagined. Our story begins not in a sophisticated laboratory, but amidst vast fields of tobacco, a plant that had become a cornerstone of economies and societies across the globe. For generations, farmers had cultivated this valuable crop, passing down their knowledge through the ages, yet a persistent threat loomed over their livelihoods, a baffling disease that could decimate entire fields, leaving behind a trail of withered, mottled leaves. The farmers had a keen eye for the subtle signs of this invisible enemy. They noticed the distinctive mosaic pattern that would emerge on the leaves, a telltale sign that their crops had been struck by the mysterious ailment. The leaves, once vibrant and green, would become marred with patches of yellow and light green, resembling an intricate mosaic. This discoloration, a symptom of the disease's insidious grip, would disrupt the plant's ability to photosynthesize, hindering its growth and reducing its yield. The farmers, dependent on their harvests, were at the mercy of this enigmatic foe. They tried various remedies, hoping to find a way to combat the disease, but their efforts proved futile. The cause of this devastating plant disease remained a complete mystery. Theories abounded, ranging from nutrient deficiencies in the soil to harsh environmental conditions. Some even speculated that the plants were being plagued by tiny insects, too small to be seen with the naked eye. At the time, the scientific understanding of plant diseases was still in its infancy. The prevailing belief was that these ailments were caused by macroscopic organisms, such as fungi or parasitic plants, or by imbalances in the soil. The concept of microscopic pathogens, too small to be seen with the rudimentary microscopes of the time, was yet to take hold. The farmers, armed with the wisdom of generations past, did their best to manage the disease. They rotated their crops, hoping to break the cycle of infection, and carefully selected seeds from healthy plants. Yet the mosaic disease persisted, a constant threat to their livelihoods and a source of endless frustration. Little did they know that the answer to this agricultural enigma lay hidden deep within the very cells of their crops, concealed within a microscopic realm that was yet to be fully explored. The stage was set for a scientific revolution, one that would forever change our understanding of the invisible world and the intricate dance between life and disease. In 1886, a German scientist named Adolf Meyer turned his attention to the perplexing tobacco mosaic disease, determined to unravel its secrets. Meyer, a meticulous researcher with a passion for plant pathology, set out to investigate the cause of this agricultural scourge. Meyer began his investigation by carefully observing the diseased plants, meticulously documenting the characteristic mosaic patterns on their leaves. He collected samples from infected fields, bringing them back to his laboratory for further analysis. Drawing upon the scientific knowledge of his time, Meyer suspected that the disease was caused by a bacterial infection. Bacteria, microscopic organisms known to cause diseases in animals and humans, were a prime suspect in the case of the tobacco mosaic disease. To test his hypothesis, Meyer conducted a series of experiments, employing the techniques available to him at the time. He crushed infected tobacco leaves, extracting the sap that flowed through their veins. This sap, he believed, held the key to understanding the disease. Meyer then passed the extracted sap through a filter designed to remove bacteria, hoping to isolate the culprit behind the tobacco mosaic disease. He reasoned that if the filtered sap could still transmit the disease, then the causative agent must be smaller than bacteria. To his surprise, the filtered sap retained its infectious nature. When applied to healthy tobacco plants, it induced the telltale mosaic patterns on their leaves, just as he had observed in the fields. This unexpected result challenged Mayer's initial hypothesis, suggesting that the disease was not caused by bacteria after all. Mayer, a careful and thorough scientist, did not immediately jump to conclusions. He repeated his experiments, each time meticulously controlling for potential sources of error. Yet the results remained consistent. The filtered sap continued to transmit the disease. Despite his groundbreaking findings, Mayer remained cautious in his interpretations. He proposed that the disease might be caused by a soluble toxin produced by bacteria, a substance capable of passing through the filter and infecting healthy plants. Mayer published his findings in 1886, detailing his experiments and proposing the existence of a filterable infectious agent. His work marked a pivotal moment in the history of virology, laying the groundwork for future discoveries that would forever change our understanding of the microbial world. Chapter 3: Ivanovsky and the Dawn of Virology. In 1892, a young Russian scientist named Dmitry Ivanovsky picked up the torch where Mayer had left off, embarking on a scientific journey that would lead to a groundbreaking discovery. Ivanovsky, intrigued by Mayer's findings, set out to unravel the mystery of the filterable infectious agent. Ivanovsky, armed with a keen intellect and a thirst for knowledge, decided to repeat Mayer's experiments, but with a crucial refinement. He employed a Chamberlain filter, a porcelain filter known for its ability to trap even the smallest of bacteria. This filter, with its incredibly fine pores, would serve as a critical tool in Ivanovsky's investigation. If the filtered sap could still transmit the disease, it would mean that the infectious agent was smaller than any known bacterium, challenging the prevailing scientific dogma. With meticulous care, Ivanovsky repeated Mayer's experimental procedure, grinding infected tobacco leaves and passing the extracted sap through the Chamberlain filter. He meticulously followed each step, ensuring the accuracy and reliability of his results. To his astonishment, the filtered sap retained its infectious potency. When applied to healthy tobacco plants, it induced the characteristic mosaic symptoms, just as Mayer had observed. The infectious agent had passed through the filter, defying all expectations. Ivanovsky, faced with this perplexing result, grappled with its implications. He realized that he had stumbled upon something truly extraordinary, something that challenged the very foundations of microbiology as it was understood at the time. He considered the possibility that the infectious agent was a new, unimaginably small type of bacterium, one capable of slipping through the pores of even the finest filters. However, this explanation seemed improbable, given the filter's ability to trap even the smallest known bacteria. Ivanovsky, a man of science and reason, refused to dismiss his findings. He meticulously documented his experiments, carefully analyzing the results and exploring alternative explanations. His findings, published in 1892, sent ripples through the scientific community. While Ivanovsky himself remained cautious in his interpretations, his work laid the foundation for a paradigm shift in our understanding of infectious diseases. He had unknowingly ushered in the era of virology, a field that would revolutionize medicine and reshape our understanding of the microbial world. Though he may not have fully grasped the magnitude of his discovery at the time, Ivanovsky's meticulous research and unwavering pursuit of truth paved the way for future generations of scientists to unravel the secrets of viruses, these enigmatic entities that continue to shape our world today. Thank you for joining us on this historical journey. The discovery of the tobacco mosaic virus paved the way for modern virology and deepened our understanding of the microscopic world. It serves as a testament to the power of scientific curiosity and the importance of challenging existing paradigms. From the farmers' observations of the mosaic-like patterns on their tobacco leaves to the groundbreaking experiments of Mayer and Ivanovsky, the story of TMV's discovery highlights the collaborative nature of scientific progress. Each step, each observation, and each experiment built upon the work of those who came before, ultimately leading to a profound shift in our understanding of the natural world. The legacy of TMV extends far beyond the tobacco fields. It has become a model organism for virology research, helping scientists to unravel the complexities of viral structure, replication, and interaction with their hosts. Stay curious and keep exploring.